Nucleic acid sequencing. Index of slides. Index of slides. Key concepts. Dideoxy-DNA sequencing. Sanger sequencing, outline. Sanger sequencing. Dideoxy-DNA sequencing. Automation of dideoxy-DNA sequencing. Maxim Gilbert sequencing. References. Key concepts. DNA sequencing is the process of determining the sequence of nucleotide bases, as TS, CS, and GS, in a piece of DNA. DNA sequencing was based on an enzymatic sequencing method first developed in the 1970s, in which a DNA polymerase was used to synthesize new DNA chains. DNA polymerase was used to synthesize new DNA chains by using a cloned single-stranded DNA template, consisting of millions of identical copies of a specific DNA sequence. Initially, DNA sequencing was slow and laborious and output was limited, but the need to scale up to conduct large-scale sequencing of library clones drove technological improvements. The DNA sequencing method that was used to sequence the human genome and many other genomes was an enzymatic sequencing method pioneered by Fred Sanger in the year 1970. It relies on random inhibition of chain elongation, creating newly synthesized DNA strands of various lengths that can be separated by size. Sanger sequencing, outline. PCR with fluorescent ddNTPS. Size separation. Laser excitation. Sanger sequencing. Sanger sequencing involves making many copies of a target DNA region. Its components are similar to those required for DNA replication or for polymerase chain reaction, PCR, which copies DNA in vitro. The components of Sanger sequencing are a DNA polymerase enzyme, a primer, which is a short piece of single-stranded DNA that binds to the template DNA and acts as a starter for the polymerase. The 4 DNA nucleotides, DATP, DTTP, DCTP, DGTP. The template DNA to be sequenced. 5 dideoxy, or chain-terminating, versions of all 4 nucleotides, DDATP, DDTTP, DDCTP, DDGTP, each labeled with a different color of dye. Sanger sequencing. Dideoxynucleotides lack the O, which allows nucleotides to join a growing DNA strand. Replication stops. Normally, this would be where another phosphate is attached, but with no hydroxy group, a bond cannot form and replication stops. Dideoxy chain termination. The blue shaded box is the site of joining. Dideoxy chain termination. The phosphate at 3-minute end has joined with hydroxyl at 5-minute end, joining a nucleotide. Dideoxy chain termination. The process repeats. Dideoxy chain termination. The process repeats. Dideoxy chain termination. Now continuing from the previous position, a new dideoxy nucleotide is to be added. Dideoxy chain termination. The violet box shows how a ddNTP is unable to form a bond with phosphorus due to lack of hydroxyl group, hence terminating the synthesis. Sanger sequencing method. Dideoxynucleotides are similar to regular, or deoxy, nucleotides, but with one key difference: they lack a hydroxyl group on the 3-carbon of the sugar ring. In a regular nucleotide, the 3-hydroxyl group acts as a hook, allowing a new nucleotide to be added to an existing chain. Once a dideoxynucleotide has been added to the chain, there is no hydroxyl available and no further nucleotides can be added. The chain ends with the dideoxynucleotide, which is marked with a particular color of dye depending on the base, A, T, C or G, that it carries. The DNA sample which is to be sequenced is combined in a tube with primer, DNA polymerase, and DNA nucleotides, DATP, DTTP, DGTP, and DCTP. The 4 dye-labeled, chain-terminating dideoxynucleotides are added as well, but in much smaller amounts than the ordinary nucleotides. The mixture is first heated to denature the template DNA, separate the strands, then cooled so that the primer can bind to the single-stranded template. Once the primer has bound, DNA polymerase will synthesize new DNA starting from the primer. DNA polymerase will continue adding nucleotides to the chain until it happens to add a dideoxynucleotide instead of a normal one. At that point, no further nucleotides can be added, so the strand will end with the dideoxynucleotide. This process is repeated in a number of cycles. By the time the cycling is complete, it is virtually guaranteed that a dideoxynucleotide will have been incorporated at every single position of the target DNA in at least one reaction. That is, the tube will contain fragments of different lengths, ending at each of the nucleotide positions in the original DNA. The ends of the fragments will be labeled with dyes that indicate their final nucleotide, A, T, C or G. Dideoxy-DNA sequencing. With the use of a primer about 20 nucleotides long, a complementary sequence is synthesized from a single-stranded DNA template. 4 parallel base-specific reactions are performed, each with all 4 dNTPs plus 1 ddNTP that competes with its dNTP counterpart for insertion into the growing chain. Size fractionation on page enables the sequence to be ascertained, giving for n plus 1 to n plus 20 the sequence GATGATGCTGAGCATTCG, the reverse complement of the sequence shown in A. Automation of dideoxy-DNA sequencing. 4 separate fluorescent dyes are used as labels for the 4 bases with ddNTP or by being attached to the primer and having 4 sets of primers corresponding to the 4 reactions. While the fragments are migrating during the electrophoresis run, a laser beam is focused at a specific constant position on the gel. The laser causes the dyes to fluoresce. Maximum fluorescence occurs at different wavelengths for the 4 dyes. The information is recorded electronically and the interpreted sequence is stored in a computer database. Example of DNA sequence output, showing a succession of dye-specific, and therefore base-specific, intensity profiles. Maxim-Gilbert DNA sequencing. A double-stranded DNA, which will be denatured to yield double strands. As the sequence of both strands are unknown, but if we find out the sequence of one strand, we would get to know sequence of other one also. At first, the double-stranded fragment is separated into 2 single strands by denaturing with high temperature or high pH. Step 1. Denaturation by heating and adding sodium hydroxide to yield single strands. Step 2. In step 2, we have to run the single-stranded fragments on gel. As lighter fragment band will move further than the heavy fragment band. How will we know which one is the lighter band? The band having larger number of purines, Ag, would be heavier. Step 2. Single-stranded DNA fragments in gel electrophoresis gel. Step 3. Now, take one of fragment band from the gel. Remove the phosphate at 5 end and incorporate a labeled one. Step 4. Radioactive labeling. Step 5. Chemical degradation. Tube 1, increase temperature and pH, by adding NaOH, that would cause fragments to break down. Dimethyl sulfate will be added that would make cuts at adenine and guanine positions. Tube 2, dimethyl sulfate and dilute HCl will be added that would cuts the fragment at adenine position. Tube 3, reagents hydrazine and piperidine are added that would cuts the fragment at position cytosine and thioamine. Tube 4, in the last tube, hydrazine, piperidine, and NaCl is added that would cuts the fragment at cytosine position. Step 6. Tube-wise fragments are produced. Tube 1 Ag cut fragments and sizes. Tube 2A cut fragments sizes. Tube 3 Tg cut fragments and sizes. 1. Tube 4C cut fragments and sizes. Step 7, Gel electrophoresis. The samples from each of the 4 tubes is loaded in the SDS page wells and allowed to run. If we look at the sequence on gel from 5' to 3', it is the exact sequence of the fragment DNA. Fragment sequence that was selected. Fragment sequence identified from the gel. 5-minute ATGCCGTTA 3 minutes. This is the sequence of DNA fragment analyzed. References: Gene Cloning and DNA Analysis, an Introduction by T.A. Brown. Molecular Cell Biology by Ladish et al.